Yeah, thank you, Greg, for the wonderful introduction and inviting me to share my research with you today. I'm very excited to speak with you. And in the next 40 to 45 minutes, I would like to share my work on using arterial spin labeling MRI to study perfusion and in applying this technique to cerebrovascular diseases. Let me see if I can forward my slide. That'd be nice. Okay. So I would like to begin by actually paying tribute to Dr. Richard Ernst, who sadly left us this month. Dr. Ernst is remembered for laying the foundation of the Fourier transform technique in MR that is now the cornerstone of tens of thousands of MR systems that enabled us to not only understand the natural world, but also saved millions of lives. As the title of my talk suggested, I'm going to speak about arterial spin labeling and how we applied ASL to conduct brain stress tests to examine the physiology of the brain. I will also discuss the principles of PET imaging and how we combine PET and MRI to integrate the strengths of both modalities. And finally, I will share some of my recent clinical studies that applied my techniques in cerebrovascular diseases. And I understand that there are a lot of imaging experts in our audience today who share similar research interests with me. So hopefully we can learn something from each other together. I would like to begin my talk by highlighting why perfusion is important to study. Now, first of all, when we talk about perfusion in the brain, we often call it cerebral blood flow or CBF. But I think it is somehow a bit unhelpful as we are not measuring flow per se, but we are actually measuring delivery instead. But because CBF and perfusion are often used together in the literature, I will consider them the same in my presentation. And also perfusion is a good metric for making comparisons between groups such as patients and controls. So this means that perfusion has become a much more popular biomarker in clinical research. And another property of perfusion is that its effect size is quite substantial. For example, this figure actually gives an example of the effect size of perfusion when our subject tapped his finger while watching a video in the MRI scanner. We can see that the perfusion change in this individual can be as high as up to 50% in certain regions. Now, equipped with these properties of perfusion, we ask ourselves how we can measure perfusion effectively. Well, actually, the same question has been was asked more than a century ago by a group in Oxford, where this is actually the first account of measuring perfusion by Dr. Charles Scott Sherrington's group, who reported some remarkable work on how perfusion responded to functional activities in the brain. So they took the measurements or the pressure of the aorta as an indication of blood perfusion and the size of the brain to illustrate functional activities. And they observed this nearly simultaneous response from the aorta and the brain to external stimulations, which implied that blood perfusion moved in a synchronized fashion with functional activities. Now, with the advent of modern imaging modalities, we can use tomography-based techniques to measure perfusion or CBF. In essence, we create a tracer using radioactive substances to monitor the kinetic of the tracer as it moves to our region of interest. Now, currently, PET imaging with radio-labeled oxygen-15 water is considered the gold standard technique for perfusion imaging. I will come back to the principles of PET and oxygen-15 water later in my talk. And of course, both DCE and DSC MRI require the injection of gadolinium contrast agents, but they are often routinely used in clinical applications. They're often used not only to report perfusion directly, but also on some related metrics, such as cerebral blood volume or the permeability of tissues and tumors. And arterial spin labeling MRI, which is the focus of my talk today, uses magnetically labeled blood water as an endogenous tracer to enable fully quantitative perfusion measurement. Now, without the need for a contrast agent, ASL can be performed multiple times in a single scanning session, which is an ideal technique for many clinical applications. Now, I'm going to use the following animation to illustrate the general principle of ASL. Now, first, we create a label in the arteries by a process of magnetic inversion. Then we wait for the labeled blood to reach the brain tissue and before we make an image. Now, at this point, we have an image of the brain with the labeled blood in it. This is known as the label image. But we also have the signal from the brain tissue itself. So we collect another image without inverting the spins, but keeping other conditions the same. Now, this is known as the control image. And the difference between the label and the control images give us perfusion or CBF. Therefore, the simplest ASL acquisition always involves two images, label and control. Now, from this demonstration, we can see that ASL relies on the subtraction of two nearly identical images. Therefore, it has a very low SNR. And also notice that the labeling and the acquisition are two independent components. So we can actually combine different MRI techniques to create the label as well as acquire the data. Now, many of my colleagues often find it confusing or difficult to understand the different ASL technology, the different ASL techniques and their acronyms, like the ones that I'm showing you here. And actually, the bigger the size of each technique, the more popular it has been used. But I always tell my colleagues that no matter how funny the names are, their goal was either to improve the quality of the ASL data or improve the accuracy of the perfusion measurement. Now, among the different variations of ASL, these are the recommendations of what is known the ASL white paper. It is a consensus from the community, and it tries to address the different ASL methods available and to give us some simple advice on what works well. And this technique has a total scanning time of about 5 minutes, and it's now available in most MRI scanners as the product ASL sequence. But it's worth noting that these recommendations were aimed at clinical applications. So it aims for something that is simple and widely available, but not necessarily trying to say what is the best. But it's a good place to start when we design our studies. And actually, in the studies that I'm going to show you today, we have chosen this recommended ASL or the standard ASL technique as the benchmark for our comparison. Now, in the earlier work of my PhD, I was quite interested in improving the data quality of ASL images. Now, because we rely on very fast imaging techniques to measure flow, the resolution of ASL data can be quite low. Now, this caused partial volume effects, where the voxel dimension is larger than the thickness of our tissue of interest, such as gray matter. Now, although partial volume effects are quite common in most imaging modalities, it can introduce severe artifacts or issues in perfusion imaging because of the unique properties of different tissue perfusions. For example, we often consider gray matter perfusion is often 3 times higher than white matter perfusion. Now, in this figure that shows the ASL signal in gray and white matter, since they have different hemodynamic properties, they demonstrate different patterns in the plot. In most clinical applications, such as studying the perfusion of dementia or cerebrovascular diseases, gray matter is often our tissue of interest. Therefore, we need to find a way to separate gray matter perfusion from the data that we collected. And the solution is partial volume correction. One approach was to estimate the gray matter perfusion by considering the signal intensities in a small region. In this technique, we assumed that the perfusion of a particular tissue, such as gray matter, is consistent in a fixed region defined by a regression kernel, such as the box in the red box here. And then we can solve a set of linear equations to determine the gray matter perfusion in each voxel iteratively. However, the disadvantage of this technique was the strict reliance of the size of the regression kernel, which could cause errors if the tissue perfusion within the regression kernel had substantial differences. Now, in order to overcome this shortcoming, we developed a more adaptive approach using Bayesian inference. In essence, our technique used a spatial prior to determine the smoothness of the data and then regulate the tissue perfusion quantification process. Now, through Bayesian theorem, the estimated values in any given voxel not only reflects the measured data, but also the prior information with the balance determined automatically, therefore making the spatial regularization technique more adaptive to the data that we actually collected. We compared our method with the linear regression approach using simulations. Now, in the ground truth, we created regions with hypo and hyperperfusion and attempted to restore them using the partial volume correction techniques we developed. Now, if you look at the regions pointed by the arrows, we can see that the linear regression technique caused blurring in the hyperperfusion region, while the spatially regularized technique was able to restore the full region of hyperperfusion. Additionally, in figure B, we created more subtle perfusion variations to simulate the perfusion variations of some diseases, for example, the early symptoms of dementia. Now, in this case, the linear regression technique nearly lost all the spatial features in the ground truth, while the spatially regularized method managed to preserve this subtle spatial variation in the simulation. Now, these data indicated that it was important to regulate perfusion quantification using the information of the data itself, which would enable us to detect subtle perfusion changes in diseases such as dementia. Another benefit of partial volume correction is improving the reproducibility of our studies. Here we are looking at the gray matter CBF of a normal subject before and after partial volume correction. And we actually collected this data from 2 sessions separated by a week. Now, without partial volume correction, the CBF maps showed substantial differences in the cortical regions on both sides of the hemisphere. But after correction, the differences was reduced because we managed to remove the errors introduced by the mismatch between the tissue thickness and the image resolution. So from this data, we concluded that it was important to use a spatially adaptive method to correct partial volume effects in order to improve the accuracy and the reproducibility of perfusion quantification. Now, as I described in my introduction, perfusion can be modulated by a variety of ways, for example, by task activities, and has a substantial effect size. So we applied this property to study how our brain perceives pain. In this study, we explored CBF variations in response to pain experiences by normal subjects. Now, it is common for us to measure the pain intensity rating to reflect the sensation of pain after we gave the participants some capsaicin, which induced a painful and burned feeling. For example, this diagram actually shows the mean pain intensity rating over time. So we hypothesized that if there was a region showing a significant coupling between the absolute CBF increase and the intensity of pain rating using this pain scale that we developed, then such region would be suitable to be a pain-specific region in the brain. So here, while the participants were experiencing different levels of pain, we collected ASL data to measure CBF. So our data showed that the CBF in the posterior insula demonstrated a significant increase over time as the subjects experiences different levels of pain. And actually, our evolution biologists also informed us that this region of the brain is actually one of the oldest regions, because the ability to detect pain has been vital for survival in the animal kingdom. Our quantitative analysis also demonstrated a strong correlation between the mean CBF and the pain rating, and a close alignment between the pain intensity and the dynamic change in CBF in the posterior insula. Subsequently, our collaborator tested the same method on pain insensitive patients, who are extremely rare and often seen to perform very extreme activities. As we expected, these patients were unable to perceive any kinds of pain during our screening, as we can see in figure A here. And then we applied the same experimental design whereby we measured their CBF in response to both thermal and capsaicin pain on their skin during imaging process. And the data confirmed our hypothesis that these patients had nearly zero CBF change during the painful stimulation, while the healthy control group showed significant CBF increase. Now, from the pain study, we not only found a specific region responsible for pain in our brain, but also explored the potential of using perfusion as a biomarker to study diseases, which inspired me to further develop this method to study other cerebrovascular diseases in my postdoc training. Now, apart from modulating CBF through task activities or a painful stimulation, it is also common to apply some pharmacological agents to alter the physiology of the brain. For example, vasodilators are some of the commonly used drugs to induce blood flow in a safe and controlled manner. For example, acetazolamide or Diamox has this off-label effect of increasing blood flow, and it can be injected through intravenous injection to our patients safely. Now, in less than 5 minutes, there's a significant increase in blood flow. And another approach is simply by asking our patients to inhale hypercapnic gas, or perhaps hold their breath for a period of time. However, this is more challenging due to the requirement of having a separate gas delivery system, or introducing some sort of discomfort to our patients. But in essence, all kinds of vasodilators targets the enzymes in the metabolism of CO2 to relax the smooth muscle cells on the walls of blood vessels to increase blood flow. Now, in clinical neurosciences, we define cerebrovascular reactivity, or CVR, to be the percentage change in CBF after vasodilation. Now, CVR reflects the capacity of the brain to respond to an external stimulation. Now, in this systematic review, we actually found that impaired CVR can increase the risk of having stroke by 4-fold. The amount of vasodilation, or CVR, can be quantitatively measured from a cerebrovascular challenge. Now, similar to the well-established cardiac stress test to reveal cardiovascular disorders, we can conduct a brain stress test to detect perfusion abnormalities that are not visible at rest, but manifest as a lack of reserve under stressful conditions induced by vasodilators. Now, since we need to perform at least 2 measurements of in a single session before and after vasodilation, arterial spin labeling is perhaps the ideal modality that enable us to perform multiple measurements of the brain in different physiological conditions. Now, as I said in the beginning, the standard ASL with a single delay was meant for measuring perfusion at a resting condition for simple clinical applications. But it was limited in the brain stress test, where we needed to image the brain at both resting and a stressful condition. The reason was that the different transit time actually affected the precise quantification of CBF. Therefore, we created a new ASL technique with multiple delays to allow mapping of the transit time at different physiological conditions. Therefore, this gives ASL more possibilities beyond measuring perfusion. Now, specifically, for the same acquisition time of less than 5 minutes, we allowed multiple acquisitions in a single TR to accommodate the increasing blood flow after vasodilation, and also included some other strategies to improve the quality of our data. We tested our multi-delay ASL on CBR measurements of normal subjects. And this diagram shows a typical design of a brain stress test where we acquired imaging data before and after vasodilation. In this case, we applied intravenously injected acetazolamide to achieve maximum vasodilation. Now here shows the CBF before and after vasodilation of a healthy volunteer. Now at baseline, the two ASL techniques nearly produced the same CBF maps, which reflected the fact that our newly developed multi-delay technique was effective in measuring CBF at resting condition and can be applied as a clinical application as well. But after vasodilation, the CBF of the multi-delay technique was significantly higher than the single delay technique. This was because the single delay technique was unable to account for the flow change induced by the vasodilator. Now, the CBF differences between these two ASL techniques were actually primarily clustered near the basal ganglia after we gave them the Diamox drug or vasodilator. This is also the location, one of the locations with the longest transit time variations. Now, without multiple acquisitions to account for the late arrival of the labeled blood, the simple standard ASL was limited to measure CBR in the brain stress test. As I described earlier, ASL provides an effective and perhaps non-invasive perfusion measurement without radiation or contrast agents. But does it really reflect the underlying vascular hemodynamics of the brain? And how does it even compare with the gold standard modality of PET imaging? So in this review, my colleague, Dr. Audrey Fan, who is actually with us today, compared different studies that applied both PET and ASL to measure CBF at baseline or resting condition. And the data showed that ASL was actually quite compatible with PET for CBF measurements, which really gave ASL a huge potential to replace PET in perfusion imaging. Now, with these promising data, we ask ourselves whether ASL can replace PET to measure CBR and potentially be used to identify patients with a higher risk of cerebrovascular diseases. But before we answer this question, I'd like to briefly go through the principles of PET imaging. So I will let you read about the etymology of PET on this slide. But essentially, it is a camera detector that detects positively charged electron coming from the patient. Now, since positrons do not exist in our body naturally, we have to introduce them through injection. Specifically, we create an unstable nucleus, such as oxygen-15, using a cyclotron. And then a proton in such nucleus decays into a neutron. And since this is an unstable neutron, it further becomes a neutrino and a positron. And now the positron travels outside the nucleus until it reaches or meets with a normal electron to annihilate. And the result of this collision or annihilation are two photons in about opposite directions. So these two photons are the exact source of signal that we detect in PET imaging. And of course, all of these events happen inside the body after we inject the radio tracer to the patient. And the PET camera can then detect millions of such events happening very rapidly. Now, to use imaging modalities to compute CBF, we often need to obtain the arterial input function, which is also the same for PET imaging. Of course, one would argue that we can do arterial blood sampling to obtain the arterial input function, but there's always the concern about the safety and comfort of our patients. So here comes the advantage of having a PET and MRI system in the same scanner, because we are able to bypass the steps of blood sampling by using our MRI data to obtain the desired AIF. Therefore, we developed a solution to derive AIF using the PET and MRI angiography. Now, using this approach, we were able to compute CBF from the dynamic PET data with deconvolution technique of the pharmacokinetic model. When I started working on this project, I wondered why oxygen-15 water PET was considered the gold standard modality for perfusion imaging. And perhaps some of you might have the same thoughts. Well, in principle, any isotopes of water can freely diffuse to tissues in the brain, which is the ideal vehicle for us to observe perfusion or delivery. And also, since the half-life of oxygen-15 is just over 2 minutes, it allowed multiple CBF measurements in a CVR study or a brain stress test, such that the signal of each measurement can decay quickly without interfering with the next measurement. However, since oxygen-15 water is still an exogenous tracer, it is considered invasive and best to be avoided. Therefore, we set out to perform a study that compared ASL and PET for CVR measurements using a simultaneous PET MRI system that can acquire both PET and MRI data at the same time. Now, in this study, we applied a similar design that used acetazolamide to dilate vessels in the brain. And we also collected different types of ASL data with single and multi-delay. Now, if we compare the two ASL results with the reference PET images, we can see that the CVR of the multi-delay method showed a higher agreement with gold standard PET than the standard ASL technique. And the reason was that the multi-delay ASL was able to capture the significant variation in transit time for different physiological conditions, which is shown in this mean transit time map of the group, that the multi-delay technique captured about 15% reduction in transit time after vasodilation. In addition, we also observed the spatial variation of transit time with the posterior region having the longest delay. Now, in many clinical applications of ASL, we always worried about the impact of ASL signal due to different flow conditions or perhaps blood compositions of different patients. Now, our data showed that the acquisition parameters that we developed were actually fairly insensitive to variations in flow and hematocrit levels, which enabled our technique to be applied to a variety of patient cohorts in different physiological conditions. Now, having developed these techniques, we investigated the potential of using ASL to identify high-risk patients with sickle cell disease, which is a genetic disorder that affects the delivery of oxygen by the red blood cells. Now, since the ability of carrying enough oxygen in the red blood cells is lower than normal for these patients, they often have a higher CBF or perfusion at the resting condition, which could potentially lead to vessel occlusion or even acute stroke due to the impaired vasodilation. Now, the current screening technique is to apply transcranial Doppler ultrasound to evaluate the blood flow of these patients. However, the disadvantage of ultrasound is its inability to identify local perfusion impairment in different regions of the brain. Therefore, we aimed to find out if ASL could potentially replace TCD ultrasound to identify sickle cell patients with impaired CBR and at a higher risk of stroke. And my collaborator, Dr. Lena Vaklovu, and I conducted this brain stress test on sickle cell patients, as well as matching controls who were the carriers of the mutated genes. Now, it was a very interesting study that these patients showed fatigue and tiredness even before the imaging session, as it took them much longer to walk from the entrance of the hospital to our scanning room. So this actually showed that they're impaired CBF. Now, perhaps some of you might recall that we shared our results in the ICP conference in 2017. Here we are looking at the CBF images before and after vasodilation. Now, for normal subjects, we could see that in the top two images, as we expected, the CBF showed a significant increase of nearly 50% throughout the brain after vasodilation. However, our patients were unable to achieve such an effect size in the same condition. This was because the CBF before vasodilation was already significantly higher than the normal range, which led to a reduced capacity for their brain to react to the stress from vasodilation. Another vascular disease that I was interested in was Moyamoya disease, which is a rare but progressive cerebrovascular disorder caused by blocked arteries at the base of the brain in the region called basal ganglia. And the name Moyamoya means a puff of smoke in Japanese. And it actually describes the appearance of these tangled and tiny blood vessels formed to compensate for the blockage, as you can see on this digital subtraction angiography. While sickle cell disease patients suffered from vessel occlusion due to the abnormality of their red blood cells, the vessel injuries in Moyamoya patients were due to the impaired geometry of their blood vessels in the brain. Therefore, we performed a pilot study to examine whether our brain stress test was effective to screen Moyamoya patients who had severe occlusion or stenosis. And also, in addition to CBF and CVR, we also aimed to evaluate whether ASL can replace gadolinium-based PWI or DSC MRI using the transit time information. So we adopted the same imaging protocol that I showed you earlier, where we applied acetazolamide as the intravenous vasodilator and performed the image acquisition and analysis using our simultaneous PET MRI system on these patients a week before their bypass surgery. And our preliminary data showed a high consistency between PET and ASL results. For example, here we are looking at a 60-year-old female patient who had occlusion in her right ACA and MCA, which we can see in the MRI angiography of the circle of Willis. Now, in stroke imaging, transit time is an extremely important biomarker to make diagnosis. We demonstrated that without contrast agent, the transit time obtained by ASL showed a high correlation with the gadolinium-based PWI technique, which indicated that ASL can potentially replace DSC as a non-invasive and contrast-free modality for stroke imaging. And of course, in the CBF and the CVR data, the arrows pointed to the perfusion deficit or the CVR deficit regions caused by vessel occlusion, in clear contrast with the unaffected regions on the other side of the brain. So these data indicated that ASL can be an effective modality to screen patients with a higher risk of stroke, such as those with sickle cell disease, as well as to identify regional CVR impairments in Moyamoya patients. Now, these data actually inspired me to believe that ASL offers a much richer information than just perfusion itself. Now, with the advent of newer and better technologies, we will be able to harvest this power and bring it more closer to our patients for longer and healthier lives. Now, during the introduction of my talk, Dr. Wheeler spoke about the podcast that I made, which you can listen to on Spotify or Anchor. And each episode is about 15 minutes and covers a variety of topics, including research, work-life balance, and career development, etc. And we are also open to new ideas and speakers. So please feel free to contact me if you are interested in making an episode. Especially, we encourage early career researchers to promote their work, as well as newly appointed faculty members to advertise your new group. And finally, if you are interested or would like to know more about my work, please follow the hashtag brain stress test. I update my research regularly on most social media platforms. I would like to thank my advisors and collaborators for making my research possible, including a lot of you in the audience today. And of course, none of my work would have been possible without the contribution of our study participants. And also, I'm thankful for the opportunity to share my work with you.